All right, great. We're almost done with our third story from Chapter 2, and therefore almost done with Chapter 2. And, of course, we want to follow up with these and bring questions to class. And we just want to point out here, all right, so we learned about gherkin, toll, the receptor, dorsal, the transcription factor that activates ventral fates. And, of course, there's pipe and there's cactus. But, on this I tried to just put the minimum, most important ones. So we still need to talk about DPP and short gastrulation. And so people don't usually say that all out all the way. Decapentaplegic, they just go DPP. So that's what I'll call it. Short gastrulation has an abbreviation that it's not such a commonly studied protein, so not that many people read it, but our book reads it SOG, which is totally legit. So you can learn these. You don't have to learn how to spell all those long words. You can just say DPP and SOG. And they're both signaling factors. They're going to be secreted from cells. And so it's going to happen in that 3-hour embryo. And so we're just going to go to this and look, okay? So in your drawing, you have that, so dorsal's bound to the twisted snail genes and it's activating them. So we got to put a little dorsal down there too, right? So that's part of your third drawing is to show, we're going to the 3-hour embryo. We're going to be down here. We're going to look at cells. We're going to look at target gene expression. And there are other target genes that are going to be regulated in these cells. Here's another one. So here's a gene that dorsal binds to, and this gene is SOG, okay? And dorsal binds to the SOG gene, and it's going to help activate it, okay? And when SOG gets activated, the protein, the SOG protein, gets secreted from the cells. And so we're going to have lots of SOG made down here because dorsal's activated down here, right? Dorsal gene is up here, but cactus is still inhibiting it. And so, we're not going to have that dorsal gene activation or regulation up here, okay? So SOG gets activated. So we could do this, you could draw basically a concentration gradient like that of SOG. So there's SOG. And developmental biologists color that in as just a triangle. There's lots down here and lots up here. And what our book does is it goes like this. It says, you do have a thing that there's a curve to this, but that's what the graph would look like, lots of it down here, a little up there, and then the same way around this other side of this bilateral symmetry like that, okay? So lots of SOG up here. It gets secreted, so, we want to show it being made and going out of the cell. And I should draw the shape like this because it's going to bind and inhibit something. So we have lots of this inhibitor that gets made up here, okay? And then there's one other gene that we have to talk about, and it's, maybe I changed the size of the pen. And the other gene you have to put in your drawing is, is DPP. So dorsal is going to bind here also. So dorsal also in your drawing. And dorsal is going to inhibit DPP, okay? And so DPP is a secreted signaling factor, and if we look up here, there would be DPP expressed up here because there's none down here because there's none down here because the active dorsal is inhibiting DPP down here. So we're going to see DPP expressed up here, and so we could do a comparable curve on your drawing like that, where there's more DPP up here because it's not being repressed. And DPP is a signaling factor. That DPP is called a dorsalizing factor. It's going to try to make the organism more dorsal. So it's a signaling factor, and it says be dorsal. And so this side is becoming dorsal. We saw down here how this side is becoming ventral, and it's becoming ventral because it's expressing ventral factors like these, and also because it's not expressing DPP. DPP is inhibited. So if you look at SOG and DPP, the final part of your drawing is you can show this, that, maybe just off to the side if you don't have space. But DPP is a protein, and it's a secreted signaling factor, so it's coming out of a cell like that, and DPP is like that, and it's trying to bind to a receptor, on a target cell. And in doing so, it's going to try to bring a signal that says be dorsal. And when it's doing that up there in the dorsal region, it's not getting to do that job because SOG binds to it. And the job of SOG is to block DPP. And this just shows how a little bit of how it's more complicated, right, than we we just talk. We just say, like, okay, dorsal got activated in the ventral. But remember how we have a variety of fates going from, where's the that slide? There. We have a variety of fates, right? Not just the most ventral fate. And so we get this gradient of fates going from the dorsal to ventral. And part of it is the dorsal protein gets activated in the ventral region and helps activate it. It also helps block dorsalizing it, right? So the ventral cells are like, you should be ventral, be ventral, and we're turning on ventral genes in you like twisted snail. We're also try to block any signal coming from the other region that says be dorsal. And so that's it. That's the end of what you need to do for the drawing. 3 drawings, and that's it. All right, thank you.